So here's the equipment as laid out for our experiment. We've got a bucket for waste, test tube rack with 4 test tubes, digital timer, petri dish labelled P containing 4 potato chips, white tile, marker pen, blunt forceps, scalpel and a 10mm syringe. Beaker with a lid containing the hydrogen peroxide, paper towels, safety goggles because the hydrogen peroxide is corrosive, and a bucket containing water for washing up. Once you've read these instructions and the invigilator says that you're allowed to open your paper, open the paper and read about the experiment. Understand it. Then we've got a set of instructions, pictorial and typed. Make sure you read and understand these. To begin with, we're going to be cutting our potatoes to different lengths as per the instructions. So I label my white tile A, B, C and D for each of the 4 different chips. Next, I take the chips. These are already cut to 4 centimeters in length. So the first one I just place by A. I will measure it just to make sure and cut it if appropriate. I take the second one, which is again measured. We see that it is in fact 4 centimeters long, but we need to cut this in half for B. So we measure 2 centimeters and we cut. Put that next to B and then work on that next piece of chip to produce C and D. Now place these back into the petri dish while we start the next part. So here are the labelled test tubes, A, B, C and D, nice and large and clear. Remembering to be careful with the hydrogen peroxide, we use the 10 cubic centimeter syringe to put 25 cubic centimeters of hydrogen peroxide into test tube A. This isn't very accurate. We'd be much better using a graduated measuring cylinder. We take chip A out of the petri dish, pick it up with our forceps, drop it into the hydrogen peroxide and start the stopwatch. We can see the hydrogen peroxide being broken down to water and oxygen bubbles. This causes the chip to rise. We stop the stopwatch when it hits the top and we record the time. The time taken is 24 seconds. Now we repeat the process for chip B, drop it in, start the stopwatch, wait for it to rise to the surface, take the time and record it, 24 seconds. Chip C, we drop it in, we can see it bubbling. It's bouncing around the bottom, so bubbles are being knocked off, so it's not necessarily rising as fast as we would think. Eventually it rises, we record the time, 1 minute 46. Chip D, we repeat the process. Again, bubbles are being knocked off so it doesn't rise as fast as we'd expect. When it does, stop the clock, record the time, 1 minute 7 seconds. Here are the results. I recorded them onto my tile. Because I've got time left, I repeat my results to get a second and third set to make the results much more reliable. As you can see, the results are a bit all over the place, but those are our results, and that's it for the prac. Not a lot to that really. Anybody should be able to do this. Right, prepare the space below to record your results. In Cambridge speak, that means draw a table. We've already pretty much done that by drawing our results onto our tile. All we have to do now then is to tidy it up a little bit and put in our units. Here are our headings: the test tube, the length of chip we were using in millimeters, the time it took for the chip to rise in seconds, and the 3 repeats, and we can do a mean as well. All that remains now then is to fill in all the details, calculate the mean, and draw in the table so that all the boxes are complete. There we are, it's complete. and that's probably 6 of the easiest marks that you will ever get from Cambridge. Okay, we've drawn the table. Now let's look for the next set of easy marks, drawing a graph. CIE always gives you a set of data to draw the graph from. In this case, we also get to do some analysis on it. In this set of results, 15 stands out, 10 and 17. They're very different from the others. Now we can calculate the mean. Leaving out the anomalous result, our mean calculates to 6.75. We stick to the same format, and so our mean is written in as 7. When drawing the graph, always do it in pencil first, and make sure you use at least 2/3 of the paper. Always remember to put the units in. If you don't, you'll lose marks. Keeping it nice and simple, 2 centimeters per pH point and 2 centimeters for every 5 seconds fits perfectly to this graph paper. When plotting, make sure you are neat and accurate. This is what CIE is looking for. So there you have it, 12 out of the possible 20 marks available for question 1, and we haven't even had to think about the more difficult questions yet.